all. I am Mrs. Subasini of the Department of Biotechnology. Today, we are going to see about biological databases, types and applications. So what are biological databases? These biological databases are storehouses of biological information. They are defined as libraries containing data from scientific experiments, published literature, and computational analysis. They provide users an efficient interface to facilitate easy recording, storing, analyzing, and retrieval of biological data through application of computer software. Now, we are going to see about the classification scheme of biological databases. The classification scheme is as follows. It is based on data type, maintenance status, data access, data source, and database design and organism. First, let's see the classification based on database design. The first one is ASN.1. It can be expanded as abstract, syntax, and notation. It comprises of a syntax. It also contains a description of how a particular data type can be represented physically in a data stream or sequential file. This is one of the database designs or file formats found in a very famous GenBank database. The advantages of this database design is its implementation is quite easy. It is a standardized design format found commonly, and the limitations in this case is the integration with the other databases is quite difficult. The next classification based on the database design is flat files. Here, the implementation is based on only one table which incorporates the complete data. Each row of the table specifies a different record. There are specific delimiters used to differentiate the records. Examples are DNA Data Bank of Japan, European Molecular Biology Laboratory. The advantages of the flat file mode of database design is it is user-friendly. The limitations in the flat file design is maintenance of data is difficult. Integration with the other databases is also difficult. There is a redundancy of data. Redundancy is nothing but repetition of the same data. The next classification based on the database design is object-oriented databases. These databases can handle complex data and they are easily integrable with object-oriented programming language. Simply, they can be defined as a collection of objects. The example of this type of a design is MitoMap. The advantages of object-oriented database design is the implementation is very easy. It supports abstract data types. However, there are some limitations too. There is a requirement for factorization of the document. The integration enhancement is also pretty difficult in this type of design. The next classification based on the database design is relational databases. These databases can be defined as a collection of relations or tables. Here, the data is organized in the form of a table where each row contains a record and each column contains an attribute of the record. Examples are SMART and GDB. The advantages of the relational database design is that is implementation is very easy. It is a reliable design. The design is quite scalable when enormous data is available. The limitations are also there. We find that there is a reduced performance when a large number of joint operations are used. The next classification based on the database design is XML. XML can be expanded as extensible markup language. This can be defined as an advanced file format, flat file format. It provides greater support for representation of complex nested data structures. It contains data definitions. It supports new definitions and tags upon requirement. The example of XML database design is found in a famous database called as SwissProt. The advantages of XML database design is It is usually fast to respond, it is better accessible, and it is quite flexible. The disadvantage is it is less mature when compared to DBMS. The second classification is based on the nature of the data stored. There are three different types of databases on this category. One is primary database, the second one is secondary database, the third one is composite database. First, now let us see about primary databases. The primary database stores data obtained from experiments. They are divided into two based on the type of data, that is, protein databases and nucleic acid databases. Protein databases contains data related only to proteins. Nucleic acid databases contains DNA or RNA related data. The primary databases are also divided into two further categories, namely sequence databases and structural databases. In the case of sequence databases, it will contain only either protein or nucleic acid sequences. Examples are EMBL, DDBJ, and GenBank. The structural databases contain protein structures only predominantly. Examples are CATH and SCOP. The second classification based on the nature of the data stored is secondary databases. These contain data derived from the primary database. Here, the data are got after thorough analysis or analytical treatment of primary data. They are further classified into metabolic pathways database, PFAM database, or protein families database. Examples are CATH, KEGG, and SCOP. Here, now we are finding three very prominent public databases. The first one is NCBI. It is also known as National Center for Biotechnology Information. It maintains a database called as GenBank. The second one is EMBL. Its expansion is European Molecular Biology Laboratory. It maintains a database known as European Bioinformatics Institute. The third one is National Institute of Genetics, and it maintains a database known as DNA Data Bank of Japan. These three databases are located in different continents in the world. NCBI is located in USA, EMBL is located in Europe, NIG is located in Japan, Asia. These databases provide free access to all the scientific researchers of the world. These contain the most updated sequences that can be seen in the genomics and proteomics research. They exchange data on a regular basis amongst themselves. In case you are uploading a data sequence or any information to GenBank, you can find the same data in EBI as well as DDBJ. The third classification based on the nature of data stored is composite database. These databases are collection of several primary database resources. This helps in lessening the tedious task of searching through multiple databases, referring to the same data again and again. The search algorithm varies considerably in every composite database. Examples of composite database are Drug Bank, Biograph, and Biomodel. Drug Bank offers details on drugs and their targets. Biograph incorporates assorted knowledge of biomedical science. Biomodel is a storehouse of computational models of biological developments. Now we are seeing the logos of secondary structural databases. The first one is CATH. The expansion is Class, Architecture, Topology, and Homology. This is predominantly a structural database containing every possible information pertaining to class, architecture, topology, and homology of proteins. The second one is also a secondary database. The expansion is Kyoto Encyclopedia of Genes and Genomes. It contains information relating to the genes and genomes of various living organisms. The third one is known as SCOP. The expansion is Structural Classification of Proteins. It contains information related to the X-ray crystallographic experiments done to elucidate the structure of the proteins. The third classification is based on the data access. That is, whether it is a public database or a private database. Public databases are small number of centers of excellence of bioinformatics worldwide. They collect, catalog, provide open access to published biological data. Examples are EMBL maintaining the European Bioinformatics Institute, NCBI maintaining the GenBank database. The DDBJ is maintained by National Institute of Genetics of Japan. The second category is private databases. These databases are maintained by a specific lab or company for their own use. Example is Celera Genomics. It is a private sequence database. This company or this genomic company was very famous during the Human Genome Project research endeavor. Here we are finding the logos of the prominent public nucleic acid genome sequence databases. The next category of the databases is based on the organisms. These databases contain exclusive data about specific organisms. Example is Flybase. The second one is SGD. SGD can be expanded as Saccharomyces genome database. For instance, Flybase contains every possible information related to the scientific experiments performed on the common fruit fly, also known as Drosophila melanogaster. In the same way, the Saccharomyces genome database contains every possible scientific experiments and information available on the Saccharomyces genomes. There is another category of databases called as specialized databases. These contain specialized nucleotide sequences like express sequence tags or sequence tagged sites. These sequences contain information relating to the chromosomal structure. They serve as chromosomal landmarks. Now we move on to the applications of biological databases. Since the explosion of the field of genomics and proteomics, the data available on the genomes of organisms have become very vast. So these biological databases have been developed to act as storehouses of information or data warehouses of organisms. These help scientists to understand the concepts of biological phenomena. These databases help in removing the redundancy of data. Removing the redundancy of data is very important for improving the clarity of the science experiments. Repetitive data tend to confuse the scientists and the researchers working on the organisms. These databases have played a very big role in removing the redundancy of the data available. These also help researchers to study the available data and form new hypotheses. They have helped immensely in rational drug design and vaccine development. They are also helpful in formulating new drugs, new vaccines, and they help in the designing of the drug targets. These are some of the applications of the biological databases. Thank you for your patient listening.